Hello everyone, welcome to today's session. And today we'll be discussing the theory related to the chapter Microbes in Human Welfare. This is the part 1 of the lecture. In this session, we'll be discussing NCERT section 8.1 and 8.2. So this is the disclaimer. So what are the objectives of today's session? So today we'll talk about the microbes in household products. Okay, which is NCERT section 8.1. Within this topic, we will understand the role of lactic acid bacteria in curd formation and its benefit in food preservation. We'll also talk about the fermentation process. Okay, fermentation is basically anaerobic respiration. And we will understand its application in common household food products like dough and toddy. Okay, we'll also analyze the microbial processes involved in different type of cheese production. Okay, so cheese is produced with the help of microorganisms. Okay, for example, Swiss cheese. Clear? So bacteria and fungus, these are used for the production of cheese. Okay, so particularly we'll talk about Swiss and Roquefort varieties of cheese. We'll also talk about microbes in industrial products, which is NCERT section 8.2. Within this topic, we'll talk about the basic principles and operation of fermenters, also known as bioreactors, in industrial microbial processes. Okay, we'll also compare and contrast the microbial processes involved in producing different alcoholic beverages such as wine, beer, whiskey, brandy, and rum. Clear? Now, we'll also explain the discovery of penicillin, which is an antibiotic. This was isolated from penicillium, which is a fungus. We'll also evaluate the role of microorganism in producing various industrial products, such as certain chemicals, okay, pharmaceutical products, enzyme, and certain bioactive molecules. So today we'll be talking about microbes in human welfare. So first I will give you a brief introduction of this topic. Then we'll talk about microbes in household products. We'll also talk about microbes in industrial products. Then we will summarize the entire session. We'll also discuss some NCERT-based questions. Okay. Then we'll discuss some keynotes that will help you memorize these topics. And in the end, there'll be some interesting questions that will enhance your critical thinking ability. Okay, let's start with today's session. So let's talk about microbes and their habitat. Where can you find microorganisms? Okay, so microorganisms, they are very tiny, they are very small living organisms. You cannot see them with naked eyes. Okay. Now, they can only be seen under the microscope. So they are microscopic living organisms. Okay. Now, they are so small, they are microscopic, but they are a major component of Earth's biological system. Okay, so they do a lot of functions. Okay, like nutrient cycling, the decomposition of the organic matter. Okay, now where can you find microorganisms? What is their habitat? Now, microorganisms, they are found everywhere. They are cosmopolitan. They are found in soil, water, air, inside our bodies as well. Okay, so they are everywhere. Microorganisms can also survive in extreme environments. So those microorganisms which can survive in extreme environment, they are known as extremophiles. Okay, and they come under the category of archaebacteria. So these microorganisms, they can survive at very high temperature, such as 100 degrees Celsius. Okay, in the thermal vents. Now they can survive deep in the soil as well. They can survive under layers of snow, several meters thick. Okay, they can also survive in highly acidic environments. These are known as acidophils. Okay, so we can say they are cosmopolitan. Okay, let's talk about microbial diversity and cultivation. Clear? So microorganisms, they are quite diverse. They include bacteria, fungi, protozoa, viruses, viroids, and prions. Here in this image, you can see different kind of bacteria and viruses. So image on the left side, you can see this image. This is different shapes of bacteria. So bacteria can be rod shaped. Okay, they can be spherical. Clear? So you can see these are the different kind of bacteria. Now the image on your right side, this is showing you viruses. Okay. Now image A, this is a bacteriophage. Bacteriophage is a virus which can infect bacteria. Okay, so this is a tadpole-like structure. Then viruses also have different shapes. Okay, so image A is bacteriophage, and B, this is adenovirus. Clear, which causes respiratory infection. Then in image C, you can see rod-shaped tobacco mosaic virus. So tobacco mosaic virus, this is a plant virus and it has RNA as a genetic material. Clear? So viruses can have either DNA or RNA as the genetic material. Most plant viruses, they have RNA as the genetic material. Now let's move on. Let's talk about the microbial cultivation. So microorganism can be easily grown on a nutritive media. So they can be grown in Petri plates. Okay, these Petri plates consist of the media, nutritive media, and their requirement is not that complex. They have simple requirements. They can grow on simple media. Clear? They can be grown on in broth also, means liquid media, and they can be grown on semi-solid media as well. In microorganisms, they form colonies, which are visible masses of them. So you can see the colonies, but you cannot see individual bacteria. Okay, you cannot see this individual microorganisms. So these colonies allow study of the microorganism. So you can study the colony. Okay, so this colony, this is, you can see a characteristic feature of a particular microorganism. So like there is one bacteria, it will form a particular type of colony. Okay, another bacteria can form different type of colony. Clear? Now fungus also forms colonies. Okay, so there's a difference between bacterial colony and fungal colony. Let's talk about bacterial colony. Appearance, they are small, they are round, they are smooth and often shiny dots. Fungal colony will be fuzzy and cotton-like with filamentous growth, because fungi, okay, their body, it is made up of hypha, and collection of hypha is known as mycelia. Clear? So that's why their colony looks like fuzzy or cotton-like with filamentous growth. Now bacteria, they reproduce very fast, they divide very fast. So the growth time is 24 to 48 hours. Clear? And fungal colony or the fungus growth time is 3 to 7 days. So what does this mean, growth time? So you can observe fungal colony after 24 to 48 hours, and you can observe fungal colony after 3 to 7 days as well, depending on the species. Here in this image, you can see in image A, this is the colony of bacteria growing in a petri dish. So you can see the bacterial colonies are round. They are small, they are smooth. Then image B, you can see this is a fungal colony. So fungus, it grows slowly. That's why it takes 3 to 7 days for the fungal colony to appear. Okay, now first let's talk about pathogenic microorganisms. So these pathogenic microorganisms, okay, they can cause disease. Okay, they are harmful. But not all microorganisms are harmful. Some are beneficial as well. Okay, many microbes cause diseases in humans, plants, and animals as well. For example, in humans, cholera is caused by Vibrio cholerae. Okay, in plants, crown gall is caused by Agrobacterium tumefaciens. In animal, anthrax is caused by Bacillus anthracis. Now, on the other hand, a vast number of microorganisms are useful to humans in diverse ways. Okay, so there are beneficial microorganisms also. So in this chapter, we will be discussing important contributions of microbes to human welfare. All right. Now let's first talk about microbes in household product. So within this, we'll be talking about curd production by bacterial fermentation. Okay. So curd is produced with the help of the bacteria, lactic acid bacteria. Example of lactic acid bacteria is lactobacillus. So they are the key players in production of the curd. They are responsible for the fermentation process that transform milk into curd. Okay. Now what is the principle? So lactic acid bacteria produce acid that coagulate and partially digest the milk protein. So milk consists of a protein, casein. Okay. Now, because of the production of this lactic acid, so lactic acid bacteria produce acid. Here, this acid coagulate the proteins present in the milk. And because of this, the milk is transformed into curd. So basically, because of this acid, the proteins are denatured. Here, the structure might change also, and the milk, it transforms into curd. Okay. Now, what is the process? A small amount of curd is added to fresh milk. So this small amount of curd acts as an inoculum because this curd consists of this lactic acid bacteria. Okay, so this small amount of curd is known as inoculum or a starter. It contains millions of lactic acid bacteria. Okay, so basically you are adding lactic acid bacteria in the milk. Okay, now at a suitable temperature, lactic acid bacteria will multiply. So for any organisms to multiply, you require optimum conditions. Clear? So at optimum conditions, the growth of that organism is maximum. So the milk is, so these lactic acid bacteria, they are producing acid. So this convert the milk sugar, lactose, into lactic acid by fermentation. Fermentation means anaerobic respiration. The acid causes coagulation and partial digestion of the milk protein, which is casein, forming curd. Okay, this is clear. So what is being done here? First of all, the lactose is being, you can say, broken down. So they are converting the milk sugar, which is lactose, into lactic acid by fermentation. This is known as lactic acid fermentation. Okay, and because of this acid, the casein, which is milk protein, this coagulates and it forms curd. Clear? Now, what are the nutritional benefits of curd? See, lactic acid bacteria enhances the vitamin B12 content, thus enhances the nutritional quality of the milk. Okay, lactic acid bacteria also enhances the gut health by checking disease-causing microorganisms. So here in this image, you can see the structure of lactic acid bacteria, and you can see yogurt. This also consists of lactic acid bacteria. Okay, example of lactic acid bacteria is Lactobacillus acidophilus. Clear? Now, second one is dough fermentation. Clear? Microorganism uses baker's yeast. What is the scientific name of yeast? Saccharomyces cerevisiae. Okay. Now, what is the principle in dough fermentation? Production of carbon dioxide by baker's yeast gives puffed appearance of the dough. So see, yeast carries out alcoholic fermentation. And in this process, carbon dioxide is being produced. Clear? So yeast is also known as baker's yeast because it is used for dough fermentation. Clear? And because of this, there is a puffed appearance of the dough because of the production of carbon dioxide. As the carbon dioxide is produced, it rises up and this causes the puffed appearance of the dough. So first of all, yeast is added to the dough. Clear? So we are talking about the process. So yeast is added to the dough used for making bread, dosa and idli. There is a question in your book. Question number 4. This is related to this chapter. So this step is very common in many recipes and is even mentioned in your NCERT exercise question. Okay. So there is a question based on this topic. Now next, what happens? The yeast ferments the carbohydrate in the dough. As it does, it produces carbon dioxide gas, which is the main reason the dough rises. Okay, clear? So what is yeast doing? It is fermenting the carbohydrates. Fermentation means in the absence of oxygen. This is kind of anaerobic respiration. Okay, and this process carbon dioxide is being produced. So basically what is happening here? Alcoholic fermentation in which carbon dioxide is produced. Now production of carbon dioxide gas causes the dough to rise and gives it a puffed up appearance. Now this process is also called leavening. Again, there is a question number 2 from this topic. Okay, now let's continue with this topic, dough fermentation. Now dough fermentation used for making dosa and idli. Okay, also used for making the bread. Okay, so in bread making also yeast is involved. Here you can see the dough fermentation process. So the dough, it consists of the carbohydrates. Okay, clear? So it consists of starch. So if you add amylase in the starch, then what happens? Amylase will break down the starch and will produce simple sugar. Okay, now yeast can use this sugar here and carbon dioxide will be produced. So basically yeast, you can say, they break down simple sugar like glucose and fructose. Clear? Sometimes yeast, they also produce amylase enzyme. So if we add yeast to the dough, yes, then what will happen? Some yeast, they produce their own amylase. So they can convert the starch in the dough, okay, to simple sugars. And then these simple sugars can be fermented. Clear? And due to this fermentation, carbon dioxide is released, which causes the dough to rise. It causes the puffed appearance of the dough. Okay, clear? So here in this image, you can see starch plus flour enzyme, which is amylase. You leave it for some time and this sugar will be produced. Okay, clear? Similarly, you just add yeast to the dough and yeast can, you can say, with the help of the enzyme amylase, can break down this starch into simple sugars and then it can carry out the fermentation process. Okay, next one is the preparation of traditional drinks such as toddy. So this is a drink that is deeply rooted in the tradition and local culture. It is a traditional fermented drink. Clear? Now it is consumed in parts of the southern India. So you can see different regions have the unique food and drinks. Okay, what is the process of preparation of toddy? The first step is preparing toddy is by fermenting the sap collected from the palm tree. Okay, now the fermentation is carried out by naturally occurring yeast. So these yeasts are present in the environment and start working as soon as the sap is collected. Now they convert, means the yeast convert the sugars in the sap into alcohol, giving toddy its unique taste and property. So what is yeast doing? It is carrying out alcoholic fermentation. In alcoholic fermentation, carbon dioxide and ethanol is produced. Ethanol is an alcohol. Okay. Other applications are used to ferment fish, soya bean, and bamboo shoots to make various food items. Okay, so this shows how versatile microorganisms are in food processing. Okay, so here you can see this is a drink which is being prepared, okay, by fermentation. Next is production of cheese. So one of the oldest milk product in which microbes were involved. So cheese is one of the oldest milk product which was prepared by using microorganisms. Now different variety of cheese are recognized by their unique texture, flavor, and the taste. Yeah, so you have different kind of cheese like mozzarella cheese, Parmesan cheese. They have different texture and flavor and taste. Now the specific characteristic of each cheese are due to different microorganisms used in their production. Okay, so microorganisms which are used for the production of these, you can say cheese. The microorganisms are producing different kind of compounds. Okay, that give cheese its flavor and the texture. Now, let's talk about cheese varieties and the microorganisms which are used for the preparation of this cheese. So cheese type is Swiss cheese and Roquefort cheese. So Swiss cheese, what are the characteristics? They have large hole because of the production of large amount of carbon dioxide. Enzyme uses Propionibacterium shermani. And then Roquefort cheese. What are the characteristics? Specific flavor. Okay, they are ripened. Microorganism use fungi, Penicillium roqueforti, growing on it. Okay. So you can see fungus and bacteria both are used for the production of cheese. Okay, cheese is also rich in protein as well. It is a good source of proteins. So cheese is a very good example of how these tiny microorganisms can have a big impact on our food production. Okay, let's talk about microbes in industrial products. Okay, so here fermenters are used for the production of these industrial products. So what is fermenter? It is a very large vessel for growing the microorganisms, for cultivating microorganisms. Okay, they can be grown on large scale. Okay, in these fermenters, the products can be obtained at large scale as well. These fermenters provide optimal growth conditions for the microorganisms. So this means we can adjust different parameters like temperature, pH, and nutrients, salts, vitamins, and oxygen as well to make sure the microorganisms as productive as possible. Okay, so if we maintain the optimum conditions, the microorganism will grow maximally. Okay, and will get the maximum product. Okay, now by creating these ideal conditions, we allow desired microorganism to produce a specific product. So this microorganism, this will, if it is growing at optimal condition, it will also produce good amount of the product. Okay, so for example, if we want to make penicillin, we use the right fungus and let it grow in a fermenter until it produces the antibiotics. This fungus is grown in optimum condition. Now, what are the significance of fermenters? First of all, fermenters are essential for large-scale industrial or commercial production. They help us turn low-value raw materials into high-value products. Okay. This is clear. Now, these devices transform raw materials into useful compounds, such as alcoholic beverages and antibiotics. For example, yeast can turn sugar into alcohol, and certain bacteria can make antibiotics. Here in this image, you can see fermenters which are used for cultivation of microorganisms. Okay, let's talk about fermented beverages. The microorganism used for the preparation of this fermented beverages is the brewer yeast. What is the scientific name of brewer yeast? Saccharomyces cerevisiae. So this yeast, this acts like a factory converting sugar into alcohol as well as carbon dioxide. So this yeast has been used for centuries in brewing and baking as well. Now, what is the principle behind this production of fermented beverages? So this process starts with malted cereals or fruit juices, which are rich in sugars. Okay. Now, when these are combined with yeast, fermentation begins. Fermentation is what is anaerobic respiration. And yeast carries out alcoholic fermentation. Now, this produces ethanol containing alcoholic drinks. So during alcoholic fermentation, ethanol is being produced. Okay. Now, the type of raw material and the processing method determines the type of the alcoholic drink. Okay. This is clear. So you can see here the raw material. If the raw material is different, then you will get different alcoholic product. So here you can see if the raw material is grape and barley, then you will get wine and beer. Okay, and wine and beer, they are produced without distillation. Okay, that's why the alcohol content is also low. Now, if the raw material is grape, barley, and sugar cane, okay, then you will get whiskey, brandy, and rum if you follow the distillation process. So by distillation, the alcohol content increases. Okay, so if you produce this alcoholic beverage without distillation, there will be low alcohol level. If you produce these alcoholic beverages by doing the distillation process, by carrying out the distillation process, then you will get more alcohol content. Okay, clear? So in the distillation process, what is done? Here what method is followed? So this method involves fermenting the raw materials, which are grape, barley, and sugar cane. Okay, and then distilling the liquid to increase the alcohol concentration, resulting in beverages with higher alcohol content. For example, whiskey, brandy, and rum. So this is an image showing you fermentation plant. Okay, now next one is the antibiotic. So antibiotics are one of the most important discoveries in the medicine, and they have saved countless lives. Okay. So what do you mean by the word antibiotic? So word antibiotic is made up of anti and bio. So this literally means against life, but in the context of disease causing organisms. Okay, so basically antibiotics, they are chemical substances produced by some microorganisms. So microorganisms, they produce a lot of bioactive compounds. They produce a lot of secondary metabolites. Okay, these secondary metabolites have bioactivity as well. Okay, clear? So these microorganisms, basically they are producing these antibiotics. Okay, and mostly they are producing these chemicals as a defense mechanism against other organisms. So these chemicals can kill or they can retard the growth of other disease-causing microorganisms. Now, what is the importance? So antibiotics kill harmful microbes that causes disease. Example is penicillin, which is obtained from the fungus Penicillium. So penicillin mainly targets and kill gram-positive bacteria, which are a specific group of bacteria with a thick cell wall. Okay, now let's talk about the discovery of penicillin. It was discovered by Alexander Fleming. What was his observation? So he worked on Staphylococcus cocci bacteria. Now, when he was working on this bacteria, what he observed? He observed a mold growing on one of his unwashed culture plates. Okay, so what he noticed that around this mold, Staphylococci bacteria was not growing. Clear? Now you can see this is an image showing you Alexander Fleming. And on the right side, you can see this is an image showing you the fungus. Clear? Penicillium. Okay, we're talking about the discovery of the penicillin. So, what was the conclusion made by Fleming? So, Alexander Fleming, he found that the mold produced a chemical that inhibits the bacterial growth. So, he named it as penicillin after the mold Penicillium notatum. So, again, there is a question in the exercise, question number 6, based on this topic. Now, what was the contribution of Ernest Chain and Howard Florey? So, the full potential of penicillin as an effective antibiotic was established much later by them. Clear? Now, antibiotics. If we were talking about antibiotics, the discovery of penicillin. Okay. So Fleming, Chen, and Florey was awarded the Nobel Prize in 1945 for this discovery. Now, what was the impact? Antibiotics have greatly improved our capacity to treat deadly diseases. Example of deadly disease are plague, whooping cough, also known as kali khansi, diphtheria, known as gal ghotu, and leprosy, known as the Kusht Rog. Here in this image, you can see Ernest, you can say Chain, and Flurry. Okay, they both of them realize the full potential of the antibiotic penicillin. Okay, now let's talk about the chemical enzyme and the bioactive molecules. So first one is organic acid. Okay, this is a table showing you organic acid extracted from the microorganisms. So acetic acid have been isolated from Acetobacter aceti, which is a bacterium. Then butyric acid have been isolated from Clostridium botulicum, which is the bacteria. Then next one is citric acid. This has been obtained from Aspergillus niger, which is a fungus. And lactic acid, this is obtained from Lactobacillus, which is a bacterium. Now the use of the significance is also given. So see, acetic acid is used in the production of vinegar. Butyric acid is also used in the production of vinegar. Then citric acid is used as a food preservative and for flavoring. And for lactic acid is used for fruit preservative and as a flavoring agent as well. Okay, let's talk about enzyme and the bioactive molecules. Okay, now what are the enzymes extracted from the microorganisms? So enzymes such as lipases, pectinases, and proteases and streptokinases, these can be isolated from these enzymes. So lipases, they are produced by bacteria such as Pseudomonas, clear, and fungi like Aspergillus niger. Clear? Now these enzymes are used in detergent formulation to effectively remove oily stains from the laundry. Similarly, enzymes like pectinases and proteases, they are sourced from microbes such as Bacillus subtilis and Penicillium italicum. Now, they are utilized to clarify bottle juice, resulting in a clearer product compared to homemade juices. Now, streptokinase, this is an enzyme derived from the bacterium Streptococcus, which serve as a clot buster in medical treatments to dissolve blood clots in patients who have experienced a myocardial infarction, which is known as heart attack. Okay, this enzyme is often produced through genetic engineering to enhance efficiency. Now, overall, these microbial products play essential role in both industrial application and medicinal treatment, highlighting the importance of biotechnology in various fields. Okay. Now, other products are cyclosporin A. This is obtained from Trichoderma polysporum. This is the fungus. Okay. This is used as an immunosuppressive agent in organ transplant patient to prevent organ rejection as well. And then statins. Here, this is obtained from Monascus purpureus, which is a yeast. Okay, so this is used as a blood cholesterol lowering agent. They act by competitively inhibit the enzyme responsible for synthesis of the cholesterol. Now let's summarize this entire session. What is the overview of the microscopic organisms are found everywhere. Okay, they are cosmopolitan. They can survive in extreme environments, and they are also known as extremophiles. So if the bacteria can survive in extreme environment, they are known as extremophiles. Now, what are the types? Okay, these can be bacteria, okay, fungi, then protozoa, and viruses. Viroids as well. Prions. Okay, these are all being considered under microorganisms. They can all cause disease as well. Okay, prions are basically abnormally folded proteins. And then viroids. Viroids are basically free RNAs. Okay, now the microorganism can be cultivated on nutritive media forming the colonies. What are the important microbial products and their applications? So see, microorganisms can be used in the household products, for the preparation of household products, for the preparation of industrial products and chemical enzymes. Okay, so in household, microorganisms are used in the production of curd, bread, and cheese. Okay, and these microorganisms involved are lactobacillus and Saccharomyces cerevisiae, which is yeast. Then for the production of beer and wine and antibiotics, Saccharomyces cerevisiae are used. Penicillin is used for the production of antibiotics, penicillin. Then citric acid is obtained from Aspergillus niger, and statins obtained from Monascus purpureus. Let's discuss some NCERT-based questions. The first question is, which of the following enzyme is correctly matched with its function? So these are the options. Pectinases remove blood clots. Streptokinase cleans oily stains. Lipases remove oily stains from laundry. And option number D, cyclosporin A, lowers the blood cholesterol. What is the correct answer? Option number C, lipases removed oily stains from the laundry. Now let's have a look at the explanation. See, lipases are specifically used in detergent formulations. Okay, now main function is to remove oily stains from the laundry. And this is an example of industrial enzyme application. Next question, a patient who has undergone heart transplant surgery would most likely to be prescribed. So options are statin, cyclosporin A, streptokinase, and pectinases. What is the correct answer? Okay, option number B, cyclosporin A is the correct answer. Let's have a look at the explanation. So cyclosporin A is an immunosuppressive agent. It is obtained from the fungus Trichoderma polysporum. It is used to prevent organ rejection in the transplant patients. Next question, the microbial product streptokinase is used as options are blood pressure medication, cholesterol lowering agent, clot buster, or detergent enzyme. What is the correct answer? Option number C, clot buster, is the correct answer. Streptokinase is produced by Streptococcus bacteria. It functions as a clot buster and it is used to remove blood clots in heart attack patients, and it is genetically engineered product. Okay, let's discuss some keynotes. Okay, we talked about the antibiotics. So discovered accidentally by Alexander Fleming in 1982. Clear? So this penicillin was discovered by Alexander Fleming. Okay, and it was isolated from the fungus Penicillium. Penicillin discovery changed modern medicine. Now Fleming, Chan, and Florey won Nobel Prize in 1945. What are the interesting facts? Okay. Our single teaspoon of soil contains over 1 billion microbes, and human body has more bacterial cells than human cells. Okay, some cheese types get their holes from bacterial carbon dioxide production. Okay, this carbon dioxide is produced by the process of fermentation. So cheese like Swiss cheese, they get their holes from bacteria producing carbon dioxide gas. A microorganism can survive in temperatures from -20 to over 100 degrees Celsius. So these are known as extremophiles. Okay, we also talk about some important bioactive molecules such as streptokinase, which is a natural cord buster from bacteria. Now, cyclosporine, this is crucial for organ transplant success. It suppresses the immune system. Then statin, this is revolutionary cholesterol-lowering drug obtained from yeast. Okay, then what are the industrial applications? Enzymes can be used in detergents like lipases. They can be used in the juice clarification like pectinases. They can be used for organic acid production like citric acid, acetic acid, and lactic acids. Here, now there are some interesting questions for you. Question number 1, I am a life-saving molecule produced by Streptococcus bacteria used to help a heart attack patient by breaking down blood cholesterol. What am I? Now, question number 2, 1928, I accidentally discovered one of the most important medicines when I noticed something strange on my unwashed culture plate. Who am I? And then option number 3, why does Swiss cheese have holes in it? So is it A, cheese develops air pocket during aging, and option B, the holes are artificially created. Okay, so you have to critically analyze these questions. Okay, so with this, we end this session. Thank you for your attention. Here, now we will discuss the DPP related to this topic. Okay, so question number 1 is which microbe is responsible for the large holes in Swiss cheese? The options are Lactobacillus, Saccharomyces cerevisiae, Propionibacterium shermani, and Penicillium roqueforti. What is the correct answer? Okay, so option number C is the correct answer here. Propionibacterium shermani. So these holes, they are produced due to the production of carbon dioxide during the fermentation process. So during fermentation, carbon dioxide is being produced. Okay, this fermentation is carried out by this Propionibacterium shermani. The next one, which microorganism is primarily responsible for converting milk into curd? The options are Saccharomyces cerevisiae, Propionibacterium shermani, Lactobacillus, or Escherichia coli. What is the correct answer? The option number C is the correct answer. Lactobacillus, which is lactic acid bacteria. This produces lactic acid. Because of the production of the acid, the milk protein casein is coagulates and milk is transformed into curd. Next one, what causes the puffed up appearance of dough used for idli and dosa? The options are production of oxygen, production of carbon dioxide, production of methane, or production of hydrogen. What is the correct answer? So option number B is the correct answer here, production of carbon dioxide. So again, this carbon dioxide is being produced by the fermentation process. So yeast is added in dough. Yeast carries out this fermentation. Okay. So the sugars are fermented, and in this process, carbon dioxide is being produced. Okay, and this carbon dioxide rises. This causes the dough to rise and causes the puffed up appearance of the dough. Next one, what role do lactic acid bacteria play in human stomachs? Options are they produce vitamin C, they inhibit pathogenic microorganisms, they digest cellulose. Option number D, they produce ethanol. What is the correct answer? Option number B is the correct answer here. So lactic acid bacteria, they are good bacteria. So they maintain the population of the pathogenic bacteria. They keep the population of this pathogenic bacteria under control. So they check the growth of these pathogenic microorganisms. Next, which metabolic pathway is responsible for carbon dioxide production in fermented dough? So options are Krebs cycle, glycolysis followed by fermentation, electron transport chain, or photosynthesis. What is the correct answer? The option number B will be the correct answer here. Okay, so what is happening here? So the glucose, which is the sugar, this is undergoing glycolysis. So with glycolysis, pyruvic acid is produced, and then pyruvic acid, you can say, it is broken down, and then carbon dioxide is being produced. The first glycolysis process is taking place. Because of this, pyruvic acid is being produced. And then yeast will carry out the fermentation process, during which carbon dioxide is being produced. Okay, this is clear. Okay, so we have discussed the DBP also. So all our answers are correct. Okay, so with this, we end this session. I hope this session was quite knowledgeable for you. We'll meet you in the next session. Thank you very much for listening.